keep glycoprotein لسه هنا. How many students attend in the first part of this lecture? Have you noticed how many students?
Yeah, I have noticed
many.
We are many.
Because I want to know if you are only 15 or 16, so I can start. But if there are more than this number, I will wait another 5 minutes in order to start.
I check while you are delivering the lecture. Those that I see, they are about 28 in number.
No, they're like 20 now.
What I have seen, I'm talking about what I have seen, not to the one that you have seen.
But they're coming. They're coming. Many are coming.
متصلين جالكسي. I will start now. I don't want to lose time, okay? Because only we have 40 minutes and the room will close again. Okay, is everything is clear that I have discussed in the previous part of the lecture? Anyone want to ask any question? Please write in the chat box. Please write in the chat box if you have any question, please. Do you hear me? Do you hear me?
Yes.
Yeah.
Yes.
Yeah,
If you have
yeah.
any question, please write in the chat box. If there is anything that you need explanation, please write it. Okay? I'm going to share the screen and to mute all students now.
Okay.
Okay. Again, we're going to speak about glycoproteins. As I said before, glycoproteins mainly composed of proteins from 50 to 90% proteins and a small amount of carbohydrate. What is the difference between glycoproteins and proteoglycan? The first difference is the nature of the sugar. In proteoglycans, which are GAGs, contains carbohydrate more than 95%. And the carbohydrate, which is polysaccharide chain, it has repeated disaccharide units. We have amino sugar attached to uronic acid. And there is a repetition of these disaccharide. Amino sugar, glucosamine, glucuronic acid, and attached to a second amino sugar, glucosamine, or a second uronic acid, attached to a third amino sugar, a third uronic acid. All these disaccharide units are linked together several hundred times to make heteropolysaccharide. But in glycoprotein, we don't have these disaccharide units. We have random sugar molecules. We may have random molecule of galactose attached to glucose, attached to fucose. We don't study fucose. You don't have to know the structure. Fucose is a monosaccharide attached to xylose, attached to glucosamine. Attached means linked by glycosidic bond, by a covalent bond. Glucosamine is an amino sugar who contains acetyl group acetic acid, or galactosamine amino sugar, galactose with amino group, and there is also acetic acid attached to galactose. N-acetyl neuraminic acid, if you remember the structure of N-acetyl, I have said its name is NANA, N-A, NA. Of course, in the exam, you will get this name. The name NA, NANA, you will get this name. You have to know what is the chemical nature of N-acetyl neuraminic acid. All these are monosaccharides or derivatives of the monosaccharides. All these sugar, they are arranged randomly. There is no repeated unit sequentially. But in GAGs, the disaccharide units are repeated in a sequence manner, and the same disaccharide unit is repeated several hundred times. But here, there is a random distribution of all of these monosaccharides: galactose, fucose, xylose, glucosamine, galactosamine, N-acetyl neuraminic acid. All these are found in glycoproteins, are found in the mucous membrane, the mucus secretion of the gastrointestinal tract, the mucus secretion of the lung, in the connective tissue. Here is also connective tissue, in the blood. Glycoprotein are found in the cell membrane, in the organelle membrane. So GAGs is found in the cell membrane, glycoproteins are found in the cell membrane. Cell membrane contains numerous structures. Cell membrane also include mitochondrial membrane, nucleus membrane, nuclear membrane, Golgi apparatus, microsomes, endoplasmic reticulum, or all the organelles inside the cell contains membrane. All these membrane contain glycoproteins. Glycoproteins may be enzymes attached to the cell membrane, may be receptors, hormone receptors, may be channels. So they play very important role. This table is found in your book, and you are supposed to study this table and to memorize it. In proteoglycan, protein content is 5%, but carbohydrate more than 95%. The major composition is usually found in the second section of the name. In the remaining part of the name, proteoglycan, the second half of proteoglycan, the second half of the name is glycan. So this is carbohydrate. This is called GAGs. The type of carbohydrate is repeated disaccharide unit. The second half of the name here is protein. This is mainly protein. You look at the second half of the name. So the protein content, it said that more than 5, it may reach to 50%. It's not 5, greater than 50%. It is mainly protein, but it is written more than 5. Carbohydrate content, mainly carbohydrate is little amount, less than 50%, but it is written here 1 to 85. Of course, there are exceptions. There are variations in the composition of glycoprotein according to the organ. You are not supposed to study the glycoprotein composition in each organ in our body. There are variations, but the majority of glycoproteins contain a small amount of carbohydrate, big amount of proteins. The nature of the polysaccharide or oligosaccharide attached, we don't have repeated disaccharide unit. We don't have repeated unit of amino sugar and uronic acid. No, there are random distribution of sugars. No repeated, no pattern of repetition. But in GAGs or proteoglycan, there is pattern. The same disaccharide unit, the same sugar is repeated several hundred times, okay? Skip. I have finished carbohydrate chemistry. I advise you to hear the record in order to understand. And I'm going to shift directly to metabolism, because metabolism is much more difficult than chemistry. As regard lipid chemistry, protein chemistry, I will make a record for them, and then you hear, and we will make a meeting to discuss what you have not understand, because the remaining time may not be enough to discuss all the module 102, all the syllabus. I will shift to metabolism because metabolism is much more difficult.
Let's see metabolism.
Okay, no problem.
Okay?
Yeah, yeah.
But we have
I'm going to send you the handout. You are supposed to study hardly the handout, but the book is very important to read in order to understand the subject. But in order to memorize for the exam, the handout is enough. We're going now to shift to metabolism, okay? Bioenergetics. نتكلم على الميتابوليزم على اللو انرجي والهاي انرجي. The first part in your metabolism syllabus is bioenergetics and electron transport chain. You will find in the YouTube channel, there are lectures in Arabic, there are lectures in English, and I will write in front of each video in Arabic or in English in order to choose your video which is suitable for you. ILOs, these are the learning objectives, and you have to, after you study the subject or the topic, you have to memorize these ILOs. You have to ask yourself, what are the high energy bonds? Examples, what are the low energy phosphate bonds? And to see if you can memorize them after you close your book, after you shut your book, if you can memorize, if you can identify the metabolic pathway, anabolic pathway, etc. Okay? So these are the topics which you should know, you should study, you should memorize at the end of each topic or chapter. What is a metabolic pathway? Metabolic pathway is a sequence of enzymatic reactions, in which the product of one enzymatic reaction enter as a substrate for the second reaction, okay? And the product of the second reaction go to the third reaction as a substrate. The product of the third reaction go and enter, join the fourth reaction at the substrate. So this is a metabolic pathway. We may have 10 or 12 steps, 12 enzymatic reactions, and all are linked together. The product of the first enter the second reaction. The product of the second act as the substrate for third. And the compounds or molecule which arises in the intermediates reaction are termed metabolites or metabolic intermediates. These are termed metabolites or metabolic intermediates. These are molecules which arises in the middle of the enzymatic reaction. As I said, we may have 10 enzymatic reactions. This is a metabolic pathway. Of course, metabolism deal with the diet in our body. Metabolism deal with oxidation of carbohydrates, which we take in the diet, oxidation of lipids, oxidation of proteins, okay? And there are other metabolic pathways. So metabolism is classified into anabolism or anabolic pathway, catabolism or catabolic pathway. Anabolism, these are biosynthesis. Biosynthesis means to make macromolecule, to synthesize macromolecules. Macromolecule to make glycogen, to synthesize glycogen from the building unit of glycogen, which is glucose, to synthesize proteins from the building units, which are amino acid, to synthesize lipids from the building units, which is mainly fatty acids. So this is the building of our tissue protein, our tissue lipid, our store of glycogen. The building or the synthesis needs energy, require energy, chemical energy. But catabolism is the degradation. Degradation means breakdown or breaking. Okay? It also involves oxidation or hydrolysis. Degradation of the large molecule, we can degrade glycogen in the catabolism to degrade glycogen into its building unit and to generate glucose from glycogen, to oxidize glucose to get energy. We can degrade proteins in our body. This occurs during starvation or fasting or starvation. Proteins are degraded into the building units, which are amino acids, and amino acids are oxidized to get energy. Also, during starvation, lipids are degraded into the building units, which is mainly fatty acids. Fatty acids are oxidized to get energy. So catabolism leads to liberation of energy, generates energy, but anabolism requires energy. All these figures are taken as a screenshot from your book. Carbohydrates, which are found in our diet, proteins from our diet, lipids from our diet, are subjected to stage 1. What is the stage 1? Is digestion. Digestion occurs in the gastrointestinal tract. Of course, digestion does not lead to generation of energy. There is no chemical energy liberated during digestion. Carbohydrates are broken into the building units, which are monosaccharides. Proteins are digested into the building units, which are amino acids. Lipids are digested into the building units, which are mainly fatty acid. Fatty acid, more than 90% of lipids. Glycerol is small amount. After digestion, the food is absorbed from the intestine, go to the bloodstream, go to the liver, go to different organs. This is a process of absorption. The stage 2 of metabolism is the beginning of oxidation. Oxidation of monosaccharides generates acetyl-CoA. Oxidation of amino acids generates acetyl-CoA. Oxidation of fatty acids generates acetyl-CoA. So acetyl-CoA is the final common pathway for oxidation of all food or diets, all constituents in our diet. Carbohydrates, lipids, and proteins all are oxidized into acetyl-CoA, has another name, active acetate. What is acetyl-CoA? It is composed of acetic acid attached to coenzyme A. Again, I said coenzyme A is one member of vitamin B complex. It's called pantothenic acid. This CoA makes chemical activation for acetic acid. So acetyl-CoA is chemically active, chemically activated by coenzyme A. Then acetyl-CoA go to two pathways. The first pathway is citric acid cycle. The second pathway is electron transport chain. So we have two metabolic pathways which generates more than 90% of the chemical energy that we generate from the diet. The citric acid cycle and the electron transport chain constitute stage 3 of metabolism, and this leads to generation of adenosine triphosphate, which is a chemical energy found in our body. Of course, oxidation generates carbon dioxide and water. Okay? This is a repetition of the previous slide. Active acetate. We get active acetate from oxidation of carbohydrate, lipid, and protein. Enter in the Krebs cycle. What is the value of the Krebs cycle? It generates reduced coenzyme. What is reduced coenzyme? Reduced means carry hydrogen. Coenzyme is the hydrogen carrier. Hydrogen carrier, this is a molecule which carry hydrogen, which are taken during oxidation of acetyl-CoA. Active acetate is oxidized, from which we remove hydrogen. Hydrogen is carried on a carrier termed coenzyme. When the carrier carry hydrogen, it is termed reduced, because reduction means addition of hydrogen. And this reduced coenzymes, which are carrier, such as NAD, niacin, vitamin B3, carry 2 hydrogen atom. NAD, coenzyme, another coenzyme is FAD, which is riboflavin, carry 2 hydrogen atom. We'll go to another pathway, which is the electron transported chain. We are going to study this electron transported chain in detail in 2 lectures, okay? And the process of oxidation of this coenzyme. What is oxidation of these coenzyme, which are NAD and FAD? Means oxidation means the removal of hydrogen. And then hydrogen meets oxygen to form water. During this process, adenosine triphosphate is generated. This process is termed oxidation with phosphorylation. Oxidation of the reduced coenzyme, removal of hydrogen from this reduced coenzyme. Of course, to write by this pen is very difficult. Oxidation. I cannot write oxidation, okay? Oxidation means the removal of hydrogen from the reduced coenzyme, is accompanied by generation of adenosine triphosphate, and this process is termed phosphorylation, means addition of phosphoric acid, linking of phosphoric acid with adenosine diphosphate to generate adenosine triphosphate. Why we need adenosine triphosphate? Why it is important in our body to generate adenosine triphosphate? Because adenosine triphosphate is used in muscle contraction. This is a chemical energy which is converted into mechanical work during muscle contraction. Active absorption of substance from the intestine needs energy. Anabolic process, I discussed anabolism, needs energy. Transmission of nerve impulse needs energy. Phosphorylation of compounds needs energy. Type, what about muscle contraction? We continuously perform muscle contraction even we are at rest, even we are sleeping, because cardiac muscles works when you are sleeping. Respiratory muscles works when you are sleeping. So there is continuous muscle contraction, continuous needs of energy. Energy in the muscle is not stored in the form of creatine, in the adenosine triphosphate. It is stored in the form of creatine. So creatine is the storage form of energy. Creatine has to gain phosphate. Will gain phosphate from adenosine triphosphate. If you look, I will go here. And phosphoric acid will be attached to creatine to generate creatine phosphate. Phosphoric acid is taken from adenosine triphosphate to sensitize creatine phosphate. Creatine phosphate is used in muscle contraction. We are going to study creatine in details in subsequent chapter, in the protein chapter, okay? When you make muscle contraction, phosphoric acid is utilized, and creatine phosphate will loses the phosphoric acid and convert it into creatine. Again, creatine should uptake or take phosphoric acid from adenosine triphosphate to regenerate creatine phosphate, and so on. This is the storage form of chemical energy in the muscle. So adenosine triphosphate is important to build creatine phosphate inside the muscle. I will stop sharing, unmute all. Is everything clear what I have discussed up till now?
Yeah.
Is my voice is clear? You hear well?
Yeah, your voice is very clear.
Okay, okay, I will continue. Okay, I will continue. حصلش ميوت ولا ايه؟ In low energy and high energy bond, you are supposed to know the definition of low energy chemical bond, high energy chemical bond, and examples by name only. Low energy bond, this is a chemical bond. Upon hydrolysis, if we hydrolyze the low energy bond, there are free energy liberated, which is less than or lower than 7.3 kilocalorie per molecule. So if we break or hydrolyze any chemical bond, there are liberation of free energy, which we can calculate. If it is lower than 7.3 kilocalorie per mole, it is termed low energy bond. High energy bond, Usually we write high energy bond like the letter S, but the letter S lie in the horizontal position, like that, letter S in the horizontal position. This is high energy chemical bond. If we hydrolyze this high energy bond, there are liberation of free energy greater than 7.3. Bigger than 7.3, it may reach up to 14.8 kilocalorie per mole. Examples of low energy bond. You can memorize example of triacyl glycerol. What is triacyl glycerol? This is glycerol, which is alcohol. We have studied glyceraldehyde in carbohydrate chemistry, but glycerol all is an alcohol attached to fatty acid. Any organic acid attached to alcohol is an ester bond. Triacyl glycerol are major lipids in our body, in our diet. Glucose 6-phosphate. If you look at glucose 6-phosphate, phosphoric acid attached to carbon number 6 of glucose. Carbon number 6 of glucose is an alcohol. So this is a chemical bond between acid, phosphoric acid, and alcohol. So this is an ester bond. Glucose 1-phosphate. Phosphoric acid is an acid. Carbon number 1 is an alcohol. So this is an ester bond. This is a low energy bond. Glycosidic bond in polysaccharides and disaccharides and oligosaccharide is low energy bond. Peptide bond which link amino acid together is low energy bonds. Okay? So you have to know examples of low energy bonds. High energy bond, you have to know examples. You are not supposed, don't study the formula. We don't require the formula. We don't require this number. This number is not required. Okay? So I will make cross. Don't study this number. But you must know an example. Enol phosphate. There is a rule in chemistry. Any chemical bonds between two acids is high energy bonds. Any chemical bond between alcohol and acid is low energy bonds. All high energy bonds are chemical bonds between two acids or groups which behave similar to acid. Okay? Pyruvic acid is an organic acid. Phosphoric acid is an acid. So this is a chemical bond between two acids. Carboxylic phosphate. Glyceric acid is an acid. Phosphoric acid, there are two phosphoric acid. Bis means two. Biphospho. We have two phosphoric acid attached to glyceric acid. Phosphoric acid is an acid. Carbamoic acid or carbamic acid is an acid. So this is a chemical bond between two acids. You have to memorize the name. Creatine phosphate, we are going to study it in great detail in protein. Adenosine triphosphate has 3 phosphoric acids. So there are bonds between 3 phosphoric acids. Adenosine di, guanosine diphosphate, guanosine triphosphate, all these are acids attached together because there are phosphoric acids. The last one is high energy sulfur. Any chemical bonds involving sulfur is high energy bond. Okay? Especially if the sulfur is attached to an acid, because sulfur is derived from sulfate, which is sulfuric acid. If you look at acetyl-CoA or any fatty acid attached to coenzyme A, coenzyme A is one member of vitamin B complex, pantothenic acid. It has sulfur. Coenzyme A has an atom of sulfur attached to the carboxyl group of any organic acid. Acetyl-CoA, acetic acid is fatty acid. Propionyl-CoA, propionic acid is fatty acid, etc. We have many fatty acids. Any fatty acid, any organic acid attached to coenzyme A is high energy bond because there is sulfur. You have here SAM, S, capital S, capital A, capital M. What is SAM? SAM, S is sulfur, A, adenosine, M, methionine amino acid. What is adenosine? Adenosine is adenine. Of course, you have studied in the high school adenine, guanine, cytosine, thymine, uracil, which enter in the structure of DNA or RNA. These are nitrogenous bases. If we link adenine to ribose, this is a glycosidic bond. Adenine attached to ribose is termed adenosine. adenosine. Okay? And this adenosine actually is attached to the sulfur bond. There is here a positive charge on the sulfur bond. Okay? The sulfur bond attached to adenosine makes high energy bond. And this compound is methionine. This is methionine amino acid. This is very important. We are going to study S-adenosyl methionine in protein metabolism, in protein chemistry and metabolism. It has many functions in our body, many biological functions. Another example is PAPS, PAPS, capital P, capital A. Another P and sulfur, sulfate. P in the organic chemistry is phosphoric acid. There is no P except phosphoric acid. Another P means we have two phosphoric acid. A, similar to A here. A is adenosine. Adenosine is adenine again, attached to ribose. And this is a glycosidic bond. This is a nucleoside. And we have two phosphoric acid. This is a phosphoric acid, very strong acid. This is a phosphoric acid. And phosphoric acid is attached to sulfuric acid. So we have two acids attached together. This is a high energy bond. Acetoacetyl-CoA. Aceto means acetic acid. This is acetic acid attached to another acetic acid. So these are two acids attached together, and this is high energy bond. And also the second acid is attached by high energy bond to sulfur. Sulfur of coenzyme A. So we have two high energy bonds. لا خلاص انا خلصت هقول لهم سلام عليكم. اعمل لهم unmute بس الاول، اعمل لهم unmute. I have finished this lecture. If anyone has any question, please. Anyone has any question in this lecture? Is it clear? أنا خلصت على فكرة.
Everything is clear.
Yeah, it is clear, but may you explain again letter B? High energy sulfate bonds? Sulfur bonds?
Okay, I will repeat it again. Wait for me for a minute, please. I will close the microphone for a minute and then I will return to you. Wait for a minute.
Fine. All right.
I will repeat the high energy sulfur bond again. Okay. طب شو بس ستوب شيرنج بس الميكروفون واضح؟ تمام كده هنعمل شير. اوكي. Any chemical bond contains sulfur, usually, mostly it is high energy bond. Because sulfur is very active molecule. It is derived from sulfate, which is sulfuric acid. If you look at this compound, مش عارفة ده ايه. لا استنى مش عارفة ايه الحكاية. ايه اللي عمل البتاعة دي؟ اوكي. Acetyl-CoA. What is acetyl-CoA? Acetyl-CoA is acetic acid attached to coenzyme A. Coenzyme A contains sulfur. This is the sulfur atom of coenzyme A. ليه خلص يا ابني ارجع. This is the sulfur atom of coenzyme A. Sulfur is attached to organic acid. Acetic acid is an organic acid. This is similar to two acids attached together because sulfur is very active. It behaves as if it is sulfuric acid. It's very active atom. Okay? You have to know the example only, but you don't need to know how and why it is high energy bond. You have to know the example. Thiol ester bond. Sulfur makes an ester bond with acetic acid or any organic acid. Propionic acid is an organic acid. It is fatty acid attached to coenzyme A. So this is an example of high energy bond. The second example is active methionine. Active means chemical active, chemical activation. Methionine is an amino acid. Of course, you are not supposed to study the formula. I discussed the formula for you in order to understand only. But you have to know the example by name only. Active methionine or SAM, S-adenosyl methionine. S is sulfur. This is sulfur atom. A, capital A, is adenosine attached to ribose. Adenosine, this is adenosine, or this is ribose. Okay? M, this is methionine. Of course, the abbreviation is very important. SAM, because S-adenosyl methionine come in the exam by abbreviations. And you are going to take the abbreviation in protein metabolism. This is an example of high energy chemical bond. The third example is PAPS. PAPS, we have phosphoric acid. This is phosphoric acid. This is another phosphoric acid. And we have also sulfuric acid. This is sulfuric acid. So we have two phosphoric acid, and this is sulfate. This is sulfuric acid. Several acids attached together, and all of them are